So AP Test loves to have you draw graphs. Make sure if you're drawing graphs, they're usually worth 3 points. And they're going to be 3 of the easiest points on the free response. You need to label your axis correctly, and that's 2 points. Each axis labeled correctly is 2 points, and then plotting to the correct points and choosing the right type of graph is another point. So think about this. Think about how many total questions the free response is. Those 3 points make up probably around 10% of the score on the free response and more. So please don't mess up the labeling. Please label. Please, please, please. All right, so we're looking at FRQ2. So there's cells. These cells like to maintain 120 millimolar of some substance L. Doesn't matter what it is. And then they put them in different concentrations of L, and they see how much is being transferred across. Like what percent is it moving? Now think about this. They're going to ask you what kind of transport is it. If it's diffusion and you put it in, is the cell going to gain or lose the millimolar? Considering it starts with 120, if you put in something with 10, it should lose it, right? But it still gains some. So right away that should tell you it's always gaining it regardless of the concentration. That's going to be an active transport. And that's why A is active transport is 1 point. Because in all of these, it should lose the substance. But in all of them, it gains it. Because if the concentration is higher in the cell than outside, and it keeps going up, it's active because you're moving against the concentration gradient. So A was worth 2 points. 1 point for saying active. 1 point for saying because it still went in the cell even though the cell had more than what was in the solution. Okay? So that was a 2-pointer. 1 point for active. And then this one asked you to explain, and you had to say because that's entry. It kept adding in L even though it should be losing it, considering it was at 120. You put it in 60, and it's still moving it across the cell membrane into itself. So it's going against the concentration gradient. Now, if it was losing, if this was the rate of losing it, that still would be weird. It would be it'd have to be reverse numbers, but then it might be passive because it's diffusing out. But it's not. It's adding it in. That silly goose. The next one's a graph. Oh, I hope this is the right thing. All right, I drew the graph here because I am lazy and I didn't want to write it on the computer. Where's my axis? I put that there. So your graph should look on number B. You get 1 point. 1 point for doing this. That's 1 point for having the correct points. Doesn't have to be absolutely precise. It just has to be relatively accurate. You get 1 point if over here you put rate of entry as a percent. As percent, because you have to label it. Just copy, honestly, on these papers, just copy. Just copy word for word what's above at the top of the table. Every time. That's what I always would do. So like you have your percent over here, copy that exact wording and put it next to your data. And down here, copy external concentration of whatever. ... But copy it verbatim with the parentheses, with the percents. Copy this onto that for your axes. That's 3 points. 1 point for this, 1 point for this, 1 point for the dots correctly. The average score on this was a 4. When they gave this, a 4 out of 10 for this question, I do believe. Or no, it would be a 4 out of 10 on normal. I think this was only worth 7 points because it's not a true AP question. But you can get almost to the passing, like getting a 4 or above would be passing just by graphing it correctly. Some things to keep in mind, remember though, for your y-axis, you can, no brakes. You cannot put brakes in it. You cannot start it. You cannot start it at like 50. You have to start your y-axis at 0. All right, you have to. It might look goofy, But it'll always fit if they give you graph paper, which they do. Like it'll always fit on there if you put the correct labels on it. Okay. So if you're having a problem getting on that graph paper, you need to look at what you're doing. All right. So that's 5 of the points. Oh, look. See, I did draw it though. Just proof I can draw. So. All right. Now, by looking at this graph, the next question on C was to determine the concentration of substance L that would be 1/2 entry rate. So 1/2 entry rate would be what as a percent? What's half as a percent? Yeah, it's 50. Thanks, Frank. I can't use you guys' real names if I publish this on the YouTube. So I go to 50. And this is actually something. And you think about at 50, where would that be on the graph? About. And, oh, yeah, it would be. So where would that, what did you guys think it was? If anyone took a guess. They accepted any answer between 30 and 34. Okay? Any answer between 30 and 34. Because you're looking at the graph, you're looking at 50%, where it is. Very common photosynthesis question. With like the color rate, they'll show the rate of photosynthesis based on the wavelength. And they'll be like, which wavelength would have this much concentration? Go to the amount of photosynthesis, go down, and that'll give you the wavelength. Okay. Again, honestly, if anyone's thinking about taking AP Stats, this is also something that we do in AP Stats. There's a lot of overlap. It's still wild to me that they have a class, like in AP Bio you do a bunch of stuff that you do in another AP class, and on top of that you also do like AP Bio. Now, that was only a 1-pointer. C's only a 1-pointer. You don't have to explain it. You could have just written 1 sentence. Don't forget you have to write it as a sentence. Complete sentences. So you'd have to put 33, or at external concentration of 33 would result in 1/2 the entry rate. Actually, they would have to accept at, you could probably just said, at external concentration of 33 MNs, you would get it. Because that's a complete sentence. Alright, now D, if you attach a giant protein to it, and remember it's going to try to enter the cell, you're making a predict. Your prediction actually should have been, it won't, it's going to have a hard time getting in. You could have said it's going to get in slower or be unable to get in. And you can say because it's too big and it doesn't fit, or because the cell will not signal with it anymore. Because it's active transport, so there's some sort of signaling chain. Both are acceptable. So that was 2 points. 1 point for saying, wait, nope. No, that's only 1 point. You just have to say likely effect. Yeah, you could just say it won't fit in anymore, or it will be unable to enter. You didn't have to say why. If they did ask you to explain to be because it can't signal in, or it won't fit in, but they didn't ask that, you just had to say it won't fit in there anymore, or it will be slower. Alright? Phew. Look at this beast. I know this sounds really nerdy, but I actually enjoy this part of teaching AP Bio because I get to read and try the questions too, and I get to actually challenge myself. Sometimes that's kind of one of the shlogs of teaching like general bio is like why you don't get this, as the memes would say. But this is actually more like I get to think as well and get things wrong sometimes, which is fun. Yeah. All right, so look at this beast. So describe the need for cells to be surrounded by membrane. All right, this one didn't have anything to do with the picture. It was asking, why is it important that cell membranes are selectively permeable? 1 point, and it's pretty much, it's so you can regulate what goes in and out of a cell. You had to say something to the extent of, you can monitor the stuff coming in those terms, regulate, pick what goes in and out. So, A, that's the answer, 1 point. And B, how does this picture show selective permeability? Well, for B, you would have to say something to the effect of these, the small lipid-soluble molecules get in without going through any doors. So they can go through the membrane, whereas in these had to go through a channel. So these can't enter in at every point of the membrane. They can only enter in specific entry points, which you can control. You can control proteins and channels. But you have some things that are allowed to go in and out freely. So this picture shows that the small lipid things go in and out freely, and then the other ones have to use a channel. And that's your answer, actually. Some enter freely, some need a doorway. And that's how it shows it. What's kind of neat about this picture is if you have another question about what kind of molecules can go into a membrane and which can't, this reminds you, oh yeah, lipids, nonpolars can go through a membrane where polar, water-soluble things cannot. That was a good one. And actually, okay, yeah. So C, describe the characteristics that phospholipid bilayer that permits hydrophobic lipids molecules to pass directly across the membrane. So all you had to say about that was phospholipids in the middle are nonpolar and hydrophobic, and hydrophobic things can pass through hydrophobic things. Polar things can go through polar things. That's actually all I had to put. Because they want to know what property of the phospholipid bilayer. Well, it's because the inside of the phospholipid bilayer is hydrophobic, and hydrophobic can go in hydrophobic, hydrophilic, and hydrophilic. So I actually could have just said the inside's hydrophobic, 100% 1 point on that. If you gave me more, that's fine. Based on the model, explain whether molecules shown across the membrane require energy. So, the big thing with active versus passive transport is active transport, you go from less to more. So like here we have 4, here we have more than 4. So if you go from 4 to more than 4, it's active because you're going against the flow. You know, there's more people in here trying to get out, trying to get in, so they're going to push their way through. Now these ones are both going from high to low concentration, so they are passive transport. So this one you would have to explain and say they do not require energy because they go from high to low concentration. Any questions? Well, here, I'll pause this if you guys. Ta-da, I did it. That's the end of the video. Uh-oh. Where's my pause button? Oh, no. Oh, no. Don't do this to me again. This is how I made a 23-hour video that one day when I couldn't get to work. Okay.